Hello and welcome to Vadose Journal. My name is Marcus Fleury and I'm a professor of soil physics and Vadose hydrology at Washington State University. I'm also the editor of Vadose Journal. Vadose Journal is a fully open access journal published by the Soil Science Society of America and Wiley Publishers. The journal is a unique publication outlet for both disciplinary and interdisciplinary research of the Vadose, the region between the soil surface and groundwater. Vadose Journal publishes both fundamental and applied research. We publish reviews, original research, technical notes, and data papers across a wide range of disciplines. A unique paper category are update papers, which are short, concise reviews of an emerging field in Vadose research. We also publish special sections, which are collections of papers originating from symposia, conferences, or workshops. The journal has an excellent editorial board consisting of the editor, 2 co-editors, and several associate editors with expertise in various disciplines of Vadose research. We have a dedicated managing staff at the Soil Science Society of America headquarters in Madison, Wisconsin. The open access mode of Vadose Journal makes all papers immediately open access for everybody to read and download from our journal website. And as all open access journals, we charge an article processing charge, but many institutions have agreements with Wiley to reduce or waive this charge. The following clips provide highlights from Vadose Journal papers presented by their authors. I hope you enjoy these short presentations. And thank you for watching.
Hi, I am Cosimo from Forschungszentrum Jülich in Germany. It is my pleasure to introduce you to my research on which I was working on together with my colleagues from the Agrosphere Institute, the University of Cologne, and the Swedish Institute of Agricultural Sciences. As a matter of fact, the rapidly growing world population, combined with recent climatic changes, pose severe food security challenges. A key to meet these challenges is a more sustainable and more productive agriculture, which can be achieved by preventing yield reduction and crop failure. A reduction in crop performance can be attributed to a number of factors, but a key one is water shortage, especially in non-irrigated regions. It is therefore essential to produce more food per unit of water. Unfortunately, our ability to simulate and thus predict the effects of water scarcity is still limited. Water stress in crop is indeed caused by precipitation patterns, but also by other factors, such as soil water content dynamics, which are in turn affected by spatially distributed soil characteristics. The simulation of those processes at the large scale is what we explore in our study. To achieve this, we focused on simulating the growth of 6 crops in 2016. This was done over an area of 1 square kilometer, where water stress and soil characteristics generally affect crop performance. Quantitative soil information was provided by a recently developed geophysics-based soil map, which divides the study area in 18 soil units. This information was used to calculate soil hydraulic parameters using a pedotransfer function. Then, using these parameters, 80 simulations of the one-dimensional AgroC model were prepared, each simulation representing a unique combination of soil and crop type. With these simulations, we investigated the soil water content dynamics and their effect on simulated crop growth. In all 80 simulations, water scarcity and crop stress were strongly related to soil characteristics as well as crop type. Higher stress occurred in coarser soils with reduced access to the groundwater. This stress affected the simulated growth of each crop by reducing their leaf area index and yield. The simulated leaf area index of all 80 combinations well matched independent satellite data. In fact, both the general crop development and the within-field patterns in crop performance were nicely reproduced. Finally, maps of the simulated agricultural yield of 4 crops were produced. Again, simulated values well matched actual harvest data or literature values. As a result, we concluded that the combination of quantitative geophysics-based soil maps with crop growth models allows precise and reliable crop simulations. This combination is particularly effective in the presence of water stress caused by soil characteristics. As a consequence, this methodology could provide a valuable support for decision-making and agricultural management. For example, specific crop types or varieties could be selected depending on soil characteristics. Also, it will be possible to investigate long-term strategies such as the best crop rotation. And finally, this methodology could make use of forecast precipitation to prescribe irrigation timing and quantity. We believe that each of these improvements can bring us one step closer to a more productive and sustainable agriculture. Well, I hope you will dig into our paper and enjoy it. To get the best out of your reading experience, I suggest you to combine this manuscript with a glass of nice Chianti from 2010 or 13. Its deep and complex flavor goes perfectly with our research topic. And remember, read responsibly. And if you read too much, ask your colleagues to drive your car. Thank you for watching this video, and don't hesitate to contact me if you have any question.
Hello, I'm Schwartz, and I would like to introduce you to the paper of my student, Tsukanov. The title of the paper is Relationship between Wheat Root Properties and its Electrical Signature using the Spectra-Induced Polarization Method. And this paper was recently published in Vadose journal. And I would like to start with some motivation. So the reason that we do this research is to study root structure, dynamic, and functions in a non-invasive fashion, or actually to develop a non-invasive method to study these important features of root. And this is important because we would like to be able to match roots to their environment, to develop stress resilience roots, and to optimize irrigation. All of that might enhance the performance of plant and increase yield. Okay, but there is some issues with that. Root research is challenging, and the main reasons for that is that first, roots are heterogeneous, really heterogeneous structures, and they are dynamic, they change in time. In addition, the growing medium, the soil, typically we don't have access to it. We cannot see what is going on there, and we need some kind of sensors to be able to sense what we have in the subsurface. Okay, so what do we do? So we do spectra-induced polarization. Spectra-induced polarization, or in short SIP, is a geophysical method that allows us to non-invasively monitor or map the subsurface or any material, actually, any material electrical properties. And it is sensitive to properties of charged surfaces. And this is important because the root cell membrane is a charged surface. The main idea with the method is to inject an oscillating current through the medium and to measure the potential. Using this type of measurement, we can calculate the in-phase conductivity, which is related to energy dissipation processes. So this is typically when we think about electrical conductivity, we think about the in-phase conductivity. The quadrature conductivity is related to energy storage processes, or in other words, to polarization. Okay, so what we did, so we grow wheat in columns and we measure the electrical properties of the wheat. We measure different zones, as you can see here in this figure. So, and what you can see here is the quadrature conductivity as a function of frequency for the different zones. And you can see that as we go down from the upper zone to the lower zone, the polarization also goes down. And here in this figure, I show the chargeability as a function of root mass, and the chargeability is the measure to the polarization, to the magnitude of polarization. And you can see that as the root mass increase, we have an increase in chargeability, and we actually have a very nice correlation between those two properties. So we have here something that can tell us how many roots we have in our medium. In addition to that, we also wanted to test the source of polarization. And for that end, we add cyanide to our system. And we did that because we know that cyanide reduced the cell membrane potential. And what you can see here is, again, the chargeability. So the measure for polarization as a function of time. And you can see that chargeability go down with time after the addition of cyanide. So we have a decrease in membrane potential, and as a result, we have a decrease in polarization. So we really can see that the membrane potential is the source of polarization for root. In addition, I show here the relaxation time as a function of time, and the relaxation time is related to the length scale of polarization. And we can see that with time, the relaxation time increase, suggesting that the length scale increase. And our interpretation is that root hairs, which those are small structures, they are affected first by cyanide. So we have an increase in the effective size that get polarized. Also, the results of our research suggest that polarization, that the relevant polarization length scale is the root diameter and not the root cell. And for this and for more detail, I invite you to go and to read the paper. So thank you very much and have a great day.